Uh, this work was done together with Isaac Overcast and Luane Landau from the California Academy of Science and from Columbia University in New York. Yes, Hoplobatrachus occipitalis is a widespread and adaptable species found across much of the sub-Saharan Africa, and they can tolerate disturbed sites, including farmlands and urban edges, and they are associated with seasonal pools and floodplain and irrigated crops. Reproduction-wise, they are associated with the onset of rainy season. And of course, of all the vertebrate groups, the amphibian species are currently the most threatened, with about 41% of them currently being threatened with extinction. And one of the problems for this is overexploitation. In Nigeria, for example, several tons of Hoplobatrachus species are sourced from the wild to meet local and international trade demand annually. And of course, locally, it's also being used to supplement protein sources of rural dwellers in the country. High level of extraction for food in this species, and together with their use in traditional medicine, would pose risk to long-term persistence of these species. We found evidences that these species are being traded, and they are sourced from the wild and pushed from the central and northern part of the country down to the southern part, to markets, in the urban markets in the southern part of the country, where our study site is located. By way of justifying our work, despite the intense harvesting pressure on this species in Nigeria, very little information on the structure, the gene flow, and diversity of the species is known. And of course, it is known that intensive harvesting may erode genetic connectivity in this species of organisms. And it has been affirmed that a need to understand the spatial distribution of genetic diversity in amphibian populations. This will definitely aid conservation efforts. And of course, the conservation genetic data that are necessary for this species and for this to take place is underrepresented for African amphibians, hence the need for this work. We set to work on this species by looking at the following specific objectives. First, we try to place Nigerian samples into a broader biogeographical context, looking at the continental view of how Nigerian samples compare with other countries within the continent. And of course, we also characterize genetic structure across Southwestern Nigeria with respect to dominant habitat types that we have across the land. And of course, we tested the relationship among the increased anthropogenic pressure and genetic diversity and gene flow. Yes, we sampled about 8 to 10 individuals per sampling site, and we have about 10 sampling sites across the whole of Southwestern Nigeria from different ecological zones. Down the south, we have the mangrove forest, followed by the rainforest, and followed by the Guinea savanna, and then the followed by the derived savanna, and then the Guinea savanna, all degrading towards the north in that sequence. And we have about 8 to 10 individuals per sampling site. Okay, when we sample these individuals, and the mitochondrial DNA and DDRAT sequence data were generated. And these were used to answer some of the questions that were used. And with 16S DNA to see how this population compare with other ones in the continent by downloading some of the the sequences from GenBank from other parts of the continent and compare. Then DDRAD dataset was also generated from 95 samples. Library were prepared, and Dankud Bank in North Carolina helped us with this, and we were able to use this to formulate and form the PCA and to look at the immigration surface of these animals and compare what this will give us. So we found out, looking at our first objective, comparing Nigerian data with that of other parts of the country, of the continent, I meant. And we found out that we have a single widespread haplotype that is representing almost all the parts of Africa, from West Africa to Central Africa to East Africa. Then we have samples from Nigeria, from Guinea savanna, from derived savanna, rainforest, and mangrove are also represented in the haplotype. And it's interesting to note that 3 unique haplotypes were found to be associated with Nigerian forests. We found 2 from the mangrove forest and 1 from the rainforest. So with the DDRAD data set, we were able to get the PCA plots. And it's interesting to note that we have a form of structuring by habitat type here. Those from mangrove forest tend to cluster, whilst those from the rainforest also tend to cluster together. We have those from the Guinea savanna that tend to cluster, and then we have those from the Guinea savanna that tend to cluster together. This is important to note because these species are widespread and seems not to structure in any way. Even though this structuring is weak, but there is a form of structuring according to habitats in this species. But there's something to note on this is the fact that a particular individual seems to cluster with another set of group. You see, this very individual is from the mangrove forest, and it tends to cluster with those from the savanna. This could be easily explained the fact that we do not actually see this as a long-term habitat association. We're seeing this as just a one-off event or a recent dispersal, because as I said earlier, trade in this species tends to come from the northern part down to the southern part. We want to believe this is just about one or few individuals escaping from captivity and be found in an environment that is not necessarily expected. Of course, more work is necessary to be able to clarify that. Yes, we try to, using our DDRAD SIG data, we were able to look at the effective migration surface for this work. And what was found out is interesting to note that there is largely a kind of geneflow, but there are some isolation in some areas. We see that there are connections among different locations, among different populations, but of course there are some striking findings. For example, if you look between Ekpe and Ikorodu, you can see that there is a form of isolation, a form of disturbance, a barrier to geneflow here. And if you look at Ibadan, and we look at Hado, and we look at Shoku and Abeokuta, also there are some forms of isolation a little bit. And I think trying to explain that, we believe what could be responsible or what is going on right now in those environments. For instance, around Epe, we discovered that there are several wetlands that are being converted now to some other use. Sand filling is taking over because what constructing roads and some other industrial houses are taking place en masse in those places. And we believe that could be responsible for what is happening between Ekpe and Ikorodu. Okay, and of course, we could explain other ones like Ado, like Ibadan and Abeokuta by saying that those places are actually state capitals. They are urban areas, and urbanization could be a reason why we are seeing some sort of barriers in gene flow in those places. We also look at the nucleotide diversity using the DDRI data set, and we felt, okay, we see a particular trend in the forest. We have more of nucleotide diversity than we have towards the north. And this can still be explained by what we're talking about the trade that has to come from the north down south. Yes, one thing is striking about Iwo, at that, there's exceptionally low nucleotide diversity in that place, which is also explainable, talking about the urbanization and aggressive farming pressure that is taking place in that place, being for its proximity to some state capital. Our conclusion, we want to say that these results indicate that although populations of H. occipitalis in southwestern Nigeria are connected in a way, certain populations may warrant targeted conservation attention due to localized pressure in this species. And sites exhibiting reduced nucleotide diversity or constrained effective migration appear most vulnerable to ongoing anthropogenic impacts. And it is important for us to maintain breeding habitat connectivity, and regulation of harvest is very important if we're going to preserve these animals for the future. And talking about the future, we felt that it would be interesting to integrate genomic-wide nuclear markers in order to be able to explain some of the things that we feel not been able to be answered in this work. And of course, to be able to, we need to also assess the evolutionary significance of habitat-associated variation, as we saw in this work. It's needed for us to be able to dig deeper and see what is really happening. And of course, it's important for us to evaluate whether continued anthropogenic pressure risks pushing this population, which has been very resilient towards genetic erosion. I want to appreciate the IFS. I want to appreciate Lakeside Fellowship. Thank you, man, for helping with data. And also I want to thank the Society for the Study of Evolution for giving me this opportunity. Thank you for listening. Hello?
Thank you. We have time for one question.
Sorry, we don't have time for a question, sorry. But
okay.
the Q&A is available down below, so feel free to put your questions in there and they can be answered anytime.
All right, thank you.
Okay, so we will move to our next speaker, Ammon Corl. Please share your slides.
Okay, can everyone hear me?
Yep.
Okay, great. Thanks everyone for coming. I'm going to be talking today about a polymorphism found in the side-blotched lizard, Judas stansburiana. And I'm assuming that people can see that. Can people see my controls?
We can just see your mouse on the screen.
Oh, okay, great. So the side-blotched lizard, is famous for playing a biological version of a rock, paper, scissors game. And in this system, there's orange males, blue males, and yellow males. And how the game works is that orange males trying to control large territories with lots of females. Blue males guard their mates closely and thereby prevent extra pair paternity. And yellow males adopt a different strategy. They don't defend a territory, but they sneak onto territories defended by other males. And as a result, orange can beat blue because of their greater testosterone and stamina. blue can beat yellow because they guard their mates closely, and yellow can beat orange because sneaking works particularly well in the large territories of orange males. And what's been observed over time is that these more frequencies cycle around. So I set out to understand the genetics of this system, to better understand how these morphs evolved and how they're maintained. And what I thought was intriguing about this is that there can these morph phenotypes, it's not just differences in coloration, it's differences in behavior and physiology and a suite of traits. And so how could this arise? So one mechanism that's been found in other systems are supergene, where typically an inversion can link to genetic variation in multiple different genes. Another possibility would be pleiotropy, where one gene can perform multiple different functions. Another possibility is we actually have multiple different genes involved in this. So previously, this system was hypothesized to be underlain by one locus with 3 different alleles, but it could be more complex than that was hypothesized. And lastly, you could have plasticity, where sometimes the same genotype can give rise to multiple different phenotypes depending on the environment. So these are some of the ideas I had going into this. So what did I find? This is showing an association between the orange and blue colors. And so it's showing basically each dot is showing a polymorphic position, and then the y-axis, the higher values are showing high levels of association. And so you can easily see that there's one single region that's associated with the difference between the orange and blue phenotypes. So this region was particularly interesting because it was upstream of a gene called cp-apterin reductase. And this gene is known to have 2 different functions in the body. So one is it makes this molecule called tetrahydrobiopterin. So here's this biochemical pathway, and SPR here is in green, and tetrahydrobiopterin is at the end of this. And that molecule is an essential cofactor for synthesizing neurotransmitters and hormones. So potentially there's a link to behavior there. At the same time, this SPR gene can help make pigments called pteridines. So cp-apterin is a yellow pigment. And also there's an orange pigment called rhizopterin, and cp-apterin reductase is acting on either precursors or directly on these different pigments. And so potentially you could have pleiotropy of this single gene linking behavioral differences with color variants. So as part of this project, I generated a reference genome for a blue morph individual and also an orange morph individual. And then I could align the 2 genomes in this SPR region. And so that's what these plots are showing. In general, the 2 genomes lined up really well and were collinear, which means that basically the genes and all the genetic sequence had kind of the same orientation at both a broad scale and a local scale. And this kind of rules out that there's evidence of an inversion in this region, which would be a change in the orientation. So we're going from like the top down to the bottom. If we zoom in even further, we can look at the alignment of, here's the blue genome and the orange genome, and gray areas are showing conserved sequence, and black areas are showing areas that are different between the 2 genomes, and lines are showing gaps between the 2 genomes. The SPR gene has 3 exons, which are here in yellow. And then so you can see that in this upstream region, it's really highly divergent between the orange and blue genomes. I also was able to do long-range PCR with primers and these 2 black arrows, and look at this across a whole variety of individuals. And it seems like even across individuals, there's multiple haplotypes, ones that tend to match this blue genome I call the blue haplotypes, and ones that tend to match the orange genome that are called the orange haplotypes. And these regions, even across individuals, there's regions that are basically unique to the blue haplotypes, unique to the orange haplotypes, which I've kind of indicated up here. And this kind of haplotype specific DNA seems to be due to transposable element insertions, which I've kind of annotated up here on the top. And so there's this highly divergent region upstream of this gene, but is only really associated with this one region, which further rules out a supergene. So As I said, I generated data for multiple different individuals here using long read sequencing with PacBio data and also with the Illumina data for the genome association study. And with that, I could finally genotype individual lizards and figure out more about what the genetic system was. And so here is a table for individuals that have 2 blue haplotypes, an orange and blue haplotype, and 2 orange haplotypes. And it turns out that all orange morphs have 2 orange haplotypes. And then for blue morphs, almost all the blue morphs that I scored had at least 1 blue haplotype. There were 3 that were homozygous for orange. I don't have photos for these 3 individuals. I can say that all the individuals that I have photos to verify their phenotype had this pattern of having a blue haplotype. So potentially these are an error. And then the yellow morphs, I haven't talked about those yet. Yellow morphs have traditionally been broken into 2 slightly different phenotypes, ones with all yellow on their throats and ones with light blue and yellow on their throats. And the ones with blue and yellow on their throats, basically all those had a blue haplotype. And ones with all yellow predominantly had a blue haplotype, but there was 4 that were homozygous orange. Kind of subsequent work looking at these individuals, it turns out these 4 individuals were relatively small compared to all the other individuals in this table. And so potentially there's an ontogenetic change where maybe individuals that are young, as they're developing orange coloration, they go through a phase where it looks yellow and then the pigments build up further. But that's something that we're going to be testing going forward. But this led to kind of a new idea about how this genetic system works. And so, as I said, all orange morphs are homozygous for the orange haplotype. The blue morphs, I think, basically, you turn a blue morph if you have a blue haplotype. And then yellow morphs, most yellow morphs are actually that blue with yellow phenotype. And so this means predominantly they'd also have basically a blue haplotype and be genetically indistinguishable from the blue morphs. And this, then how do you get these different phenotypes? Well, there's been some field observations that suggested that yellow morphs can transform to blue morphs if a territory opens up. And so the idea here is basically phenotypic plasticity could give rise to these 2 different phenotypes with the thought that basically if you're a male and you don't have a territory, you stay yellow and you sneak. But if you can get a territory, you turn blue and you guard your mates. So this Cyblotch lizard system is kind of well known for how it maintains variation through a rock, paper, scissors game. So I wanted to basically revisit the models with this new genetic idea and hypothesis in mind. And so I've kind of talked about the framework for how I would break up the genetics of it. And then for the plasticity, initially what I did was I modeled it where basically yellow morphs would be a constant proportion P of the individuals with a blue haplotype. And then blue morphs would be just a constant 1 minus P proportion of individuals with that haplotype. And I kept that constant across different generations as a simplifying assumption. I'll get into how I changed that. And then the relative fitness of each of these morphs was based on payoff matrices that had been previously estimated from the field data. Okay, so the cool thing about this was that even this relatively simple genetic system where you have 2 different alleles and then plasticity giving rise to a 3rd phenotype, they can still play rock, paper, scissors. And this rock, paper, scissors game can still maintain that genetic variation. And so this is looking at the allele frequency over generations on the left and the phenotype frequency over on the right. And this is just one iteration of this model, but it shows that you can maintain variation going forward with this simple genetic model. However, as I mentioned, there's been observations in the field of basically yellow morphs transforming to blue morphs within a breeding season when a territory opens up. And so I was trying to think about how I could incorporate this variation within a generation. And so what I came up with was I could calculate the optimum proportion of blue and yellow morphs, which is simply when the fitness of blue morphs equals the fitness of yellow morphs. And I also explored when that proportion deviated from the optimum within a generation. So basically different generations would have different proportions depending on what was going on. And then what was interesting about these particular models is that some of them exhibited cycles in the allele frequency and the phenotype frequencies. And it gave rapid cycles kind of similar to what's been observed in the field, where in the field, the morphs are cycling about every 5 years in frequency, and these models are sequencing every 6 generations. And what's also interesting is that when I had fixed that proportion of yellow morphs in the previous models, none of those exhibited cycling behavior. So adding this extra plasticity kind of potentially gives more biological realism. And then lastly, I took all these models and I tried to simulate them with genetic drift to look how stable this system of polymorphism is maintained. So I basically measured the proportion of 1,000 trials in which the polymorphism is maintained for 1,000 generations. So the y-axis here is going from kind of unstable to stable at values of 1. And the gray line here is that optimized model where the proportion of blue and yellow morphs can change within a generation. That turned out to be have the highest stability of all the models. And then the different colored lines are showing ones with a constant proportion of the blue and yellow morphs. And so some of those actually have higher stability than the previous 1 locus 3 allele model. So it seems like plasticity can lead to increased stability in the system. And so just to summarize all that, the lizard phenotypes seem to result from genetic variation at this SPR gene. And this cypriapterin reductase gene also convergently generates color morphs in a distantly related lizard called the European wall lizard. The morph phenotypes are likely to rise because of pleiotropy, not because of a supergene. And supergenes are a common mechanism for generating morphs, but this kind of shows that variation in a key regulatory gene is sufficient for generating these highly distinct morph phenotypes. And lastly, these morph phenotypes likely arise from just only 2 alleles, with plasticity giving rise to a third phenotype that can generate a rock, paper, scissors game. And basically, even the simple genetic system can basically maintain variation going forward and can do so particularly stably, and plasticity kind of enhances that. And so it's a new way of thinking about both balancing selection in this system, but also balancing selection more generally. And with that, I'd like to thank the large number of people who helped do this project, but especially Rasmus Nielsen, whose lab I did this work in, and Barry Sinervo, who is my long-term collaborator, who really developed the rock, paper, scissors game and the side blotch lizard. There's now a publication about this, and feel free to contact me if you have any other questions.
I'm here to present my work in the gene expression evolution in salivary glands. So I'm interested in how gene expression is involved in the generation or the evolution of phenotypic differences. And I think this is a question that as a field we have been studying for at least 50 years since this beautiful paper that is like a fundamental paper in which they suggested that gene expression makes up for a lot of the phenotypic differences between humans and chimpanzees. And the question, I think, is how do we study or how do we measure the evolution of gene expression across species? And there's multiple ways of doing it. Mainly, we try to compare across species and try to find patterns and see if there's something that comes out as different as the pattern in the other species. Then we can look at that and see if that could be related to adaptive evolution. And people have done this, and they have looked at gene expression across multiple species and multiple tissues. And the idea that the idea that comes from these studies is that basically the gene expression should cluster within tissues more than across species and across sexes. So the idea is that, for example, a human liver will be more similar to, let's say, a dog liver than it would be a human liver to a human brain, right? And we can see that across multiple comparisons. And so that makes sense because the liver will have a common ancestor, for example, in all of these species, and then it needs this group of genes that will perform very similar tasks and that would be conserved across evolutionary time. And so we also know that there's different levels of variation in gene expression across species. And for example, the testis is known to be one of the tissues that mostly vary as compared to other tissues, for example, as brain that varies the least, or in these studies, for example. And so I hope with this talk to show you how interesting the salivary glands are to understanding the evolution of gene expression. And the salivary glands are these 3 glands that all mammals have, and they secrete proteins into our saliva. That these proteins are related to everything that happens in our mouth, digestion, immunity, oral health, so on and so forth. And they're mainly different a little bit by function. So parotid would be more related to digestion, and then submandibular and sublingual more oral health and the viscosity of your saliva, for example. And so previous work in our lab, we were studying this locus called the SCPP locus that has genes that are related to actually multiple, that are expressed in multiple different tissues. But the ones that were highly expressed in saliva in humans, we didn't find them highly expressed in saliva in mouse. Or we found them, but it was not exactly the same ortholog, or there was differences in gene expression. So that motivated us to explore a little bit more of like, what are the differences in gene expression between human and mouse in the salivary glands, to see how that evolved. And so what I did in this project is we sequenced the RNA-seq for 3 glands: parotid, sublingual, submandibular, and pancreas and liver. And we looked at, we compared this with gene expression data that is available for humans. And we try to understand a little bit of what's going on in these glands. So this first image, to me, was quite unexpected. So like I showed you before, we expect a lot of conservation in the genes across tissues. So I would expect to see the top genes, or most of the genes that are that are expressed in human glands, to be also expressed in mouse glands. And so in here, this figure shows we filtered data set for genes that were secreted, or it's genes that code for proteins that are secreted in saliva. So we wanted to have a look at understand what's going on in saliva. And then here I have the bubble sizes are proportional to the gene expression, and the colors reflect orthology. So for example, in parotid, and the top is a mouse and the bottom is human. So for example, in parotid, we see that a lot of the genes are green. So these are 1-to-1 orthologs. So meaning is that if the gene is present in mouse, it's also present in humans. You can see my mouse, right? Hopefully you can. However, for submandibular and sublingual, a lot of the genes are lineage specific. So these are genes that are highly expressed in mouse, but are not present in humans. And that called a lot of our attention. We didn't find any other description of this in the literature. And so we were very motivated to look more into this and what's going on, that these are like specialized by species, or something that is going on here. And so another way into looking at this, is looking at total gene expression. So in the y-axis here, we have gene expression for secreted proteins, the proportion of gene expression for secreted proteins that makes up for each one of these orthology groups. So in parotid, we see that, at least in mouse or like in humans, is like more than 60% of the genes are 1-to-1 orthologs. But when we look at submandibular and sublingual glands, most of the genes that are expressed in these glands are lineage specific genes, are genes that are present in mouse and not in humans. And so that was very surprising to us, and it was a little bit different from what we saw in the literature, and was interesting. So we validated this to see, because we wanted to see, okay, does this reflect the actual proteome of saliva? And so what we did is we got we sequenced the proteome in saliva, And I correlated the protein abundance with gene expression abundance for each of the salivary glands. Here I'm only showing submandibular, but all of them kind of look very similar. So they're all positive correlated with the abundance in saliva, of protein abundance in saliva. And especially if we look at the top genes, which are the guys that I showed you before, we see that there's higher correlation, meaning that if these genes are highly expressed in these glands, they're probably also highly abundant in saliva. So that kind of validates that this is something that we actually see in saliva, and that's probably something functional related to this. The next thing we ask is, we know that in the submandibular gland, there's sex differences in mouse. So the salivary glands have this structure, secretory structures, in which they have the duct cells and the acinar cells. And males have more of these structures as compared to females. So we wanted to see if that reflected also in the transcriptome. So what we did is we did differential gene expression between females and males for all the salivary glands, pancreas, and liver as comparison. And what we found is that in the submandibular gland, there's a lot of genes that are differentially expressed. So almost more than 1,500 genes are differentially expressed. And this was, again, another surprising result, because we know that liver is already sexual dimorphic, and it is at least 5 times less than submandibular gland in terms of genes that are sexually dimorphic. So this was very interesting, and we wanted to explore a little bit more, like what are the genes that are actually making up all of these genes, and what could be the functions related to it. And so we looked at a human too, to see if there was some differential expression in submandibular gland, and there's not much. Most of these genes that you're seeing here are Y chromosome genes or X chromosome genes. So it was something very unique to mouse in this case. And we saw a lot of genes that are highly expressed in males. So there's something functional here, or like at least that was my hunch. So we looked at these genes more closely, and we found this very interesting family of genes that are all highly expressed in males, which are the kallikrein genes. So the kallikrein genes make up for closely to 16% of all expression in submandibular glands of males. And so they're a very important part of the gene expression of this gland, and most of them are highly expressed in males. So I wanted to see if how are they in the genome? Are they clustering together? And what we found when we compared to humans is we saw that all of these 1B genes here that you are seeing, all of these genes are mouth specific. Meaning that they were duplicated in the mouse lineage, and they all have male-specific expression. They're also clustered in the genome. So all these white circles here are pseudogenes. They're in the middle, but all of them are actually one after the other. And so I thought this was very interesting, and there has to be some regulatory mechanism that is starting the expression for all of these genes together. And so my idea was to look to see if there was any motive that could be, for example, a transcription factor binding motive that could regulate all of these genes together. So I was trying to look at something that could explain how all of these guys are highly expressed in males. And so I did some de novo motive analysis that looks for motifs that are common across all of these genes. And we found one motif that serves as a transcription factor binding motif for this protein called GHRL2. And we know that from the literature that GHRL2 is present in the presence of testosterone. So that gave me a hunch that there's some hormonal regulation of all of these genes together. That was really cool. And then we went a little bit further with this. So I wanted to see if there's any 3D genome structure that could also explain this pattern. And I looked into Hi-C data. So basically it looks like how close two regions on the genome are together. And we found that there are 2 TADs in the region. So these TADs are these chromatin loops that mostly indicate that these genes that are within the loop are being co-regulated. And the TADs overlap exactly where the genes where the boundaries of these genes are. So the idea is that these genes, all of these genes are male specific. They are they're co-regulated probably by the same transcription factor, and they're within the same TAD. So that explains how they're being co-regulated. And so we built this model to kind of understand everything that I said, in which there was somehow in the evolution of the mouse lineage, there was a gene duplication that acquired a motive, that acquired a function, because then there's some hormonal regulation of the expression of these genes. And then there's this duplication occurred within a TAD. And then that led to the difference in sex, in expression between the sexes. And so that was what our kind of model to try to understand how this phenotypic difference that was the highly expression of these genes in the mouse salivary glands occurred. And hopefully I could give you a little bit of an overview of like how salivary glands could be this interesting model to understand gene expression evolution, because salivary glands, we think salivary glands are evolving a little bit faster because of it needs to respond to different diets and environments faster, maybe than other tissues, but we still have to test this hypothesis. And so this is one preliminary idea of like how this could be an interesting model. And so I told you that there's a lot of diversity in gene expression in salivary glands, and then there's some genes that are also related to sex-specific phenotypes that we can look at in mouse. And lastly, I just wanted to say that I didn't talk about the function of the kallikrein genes because I didn't want to go into that. There's a lot of things in the literature, but they are proteases, and they are also known to be related to tissue repair. And so it might be that the male mouse are licking themselves after they had a fight or something that could be explaining why they're highly expressed, but this is just a hand-wavy hypothesis. And with this, I just wanted to thank you everyone for listening to me and my lab mates and collaborators, and I would be happy to take any questions if there's time.